So welcome to today's Immune Zoom seminar. And this is also our launching seminar for this summer series. So starting from today, we will have two seminars each week. So one usually in the evening of Saturday, and the other is in the evening of Wednesday for US time. Today it is my absolute pleasure and honor to have our speaker, Dr. Dolly Mehta, join us, who is currently a professor at the University of Illinois in Chicago. Dolly did her college in Rockingham University in India and a PhD in Delhi University. She received a postdoc training in Indiana University. In 2004, Dolly started her own group as an assistant professor in the Department of Pharmacology at the University of Illinois, Chicago, and was promoted all the way to the full professor. Dolly's research focused on lung homeostasis and regeneration. She attempted to address one of the key questions in the field, which is how the decision is made between inflammation versus resolution following lung injury. Her lab made a few seminal discoveries, which include the identification of role of sphingosine 1-phosphate receptor 1 in restoring endothelial permeability after injury, the regulation of restrictive endothelial fate by focal adhesion kinase, and the mechanism of how GPCR part 2 dampens inflammatory signaling in alveolar macrophages. So there's a long list, and I will not read all of them because I don't want to take up too much time for Dolly's talk. So today, Dolly will share with us about her recent effort in understanding macrophage plasticity and lung injury. So without further ado, Dolly, the floor is yours.
Thank you, Charlie, and Dr. View for inviting me to speak. And before I start discussing about macrophage plasticity and lung injury, I would like to acknowledge the fellows who have done the work, the current fellows and the alumni, and the collaborators within UIC, University of Calgary, and China, who have provided resources and sometimes experiment data to really help us understand what is this macrophage plasticity happens during lung injury and how they behave. So I think everybody knows about this alveoli, which are a small balloon in the lung. And right now everybody is sitting and hearing me because your lungs are dry, which is this is healthy alveolus. And if you can see that this alveolus, healthy alveolus, has alveolar macrophage, and then type 2 epithelial cell, and then type 1 epithelial cell, and this is the capillaries. That is the major barrier. So nothing goes into the alveolus because it acts as a barrier. And in a general sense, these alveolar macrophages respond to any pathogens because lung is in direct contact with the environment. So what happens is it can take all these alveolar macrophages, according to Paul Kuop's data, they can take care of pathogens build up to a PFUI to about 10 to the power 4. But if this pathogen load increases, they call for neutrophils, and neutrophils comes from the opening of the junctions between the endothelial cells. And these then neutrophils get activated because alveolar macrophages are trying to secrete inflammatory cytokine. And that in turn lead to, when the endothelial cell open, to allow neutrophils, that also allows for protein-rich fluid accumulation, and this happens in the injured lung. But if this injury is sustained, what happens is that you will get irreversible injury of the alveolus, and that has caused lethality in patients of lung injury, which we have experienced in past 3-4 years, leading to mortality for COVID. So we are trying to understand how we can train alveolar macrophages so we can reverse this injured alveolus and facilitate gas exchange in the sick patients. And that's the whole motto of the lab research on the fellow, which are working on the macrophages. So we know that I mentioned about the alveolar macrophages, but we also have interstitial macrophages in the lung. And recently, Claudia described that these interstitial macrophages are of different flavors and perform different functions. So there is so much complexity of these macrophages, what they become and how they originate and what are their functions. So we are still trying to understand at what stage these macrophages can be called purely as alveolar macrophage or interstitial macrophage. And there I will describe in next slide, there are also monocyte-derived macrophages. So where these alveolar macrophage originate after injury, we know that alveolar macrophages develop in the lung from different source as compared to monocyte-derived macrophages or interstitial macrophages. But when you have injury, the recovery has to happen. And during injury, most of the alveolar macrophages die. We know about 80% of these macrophages either die or they somewhere go, which we are not able to understand where they go. Then there is also interstitial macrophages, which can undergo some kind of a phenotypic switch to form alveolar macrophages. But there are also studies that these alveolar macrophages proliferate to reinstate the lung homeostasis. However, there are several studies showing these monocytes can become macrophages and they could attain a function of alveolar macrophage-like function. There they can work with alveolar macrophages, which are proliferating to provide a niche to restore the lung homeostasis after injury. So what are these macrophages? That's the topic as I described. And what we have shown initially in a couple of studies in past 4 or 5 years, that when you hit the lung with bacteria such as Pseudomonas aeruginosa or lipopolysaccharide or LPS, which are the two very common pathogens that are affecting the lung injury patients in the clinical setting. So what we have shown that these two, even any injury can release thrombin from during injury because of there is lot of hemostasis disturbance and platelets can release that. And that this thrombin can ligate the receptor which are called protease activating receptor 1 and receptor 2, part 2. So these are G protein coupled receptor, while this is the TLR4. And we know that this TLR4, and Charlie and Sean has published a lot, that these TLR4 can activate NF-kappa B signaling and regularly induce the inflammatory cascade by generating IL-6, IL-1 beta, and TNF-alpha. But we've found that when you have this activation of PARs, there is a yin and yang between this PAR1, TLR4, and PAR2 receptor. And what we have shown here is that when you don't have part 2, there is a exaggerated lung injury because this part 2 is producing cyclic AMP cascade. And this cyclic AMP then blocks calcium coming from TRPV4 channel. And what we showed in this paper was this calcium was activating phosphorylation of NFAT by calcineurin. And this NFAT somehow augmented the NF-kappa B activity. So if we take away this pathway, if we inhibit cyclic AMP release, this pathway get exaggerated and there is a sustained injury. But when we provide part 2 back into these mouse, which were part 2 null mouse, only in the macrophages, we could show that this injury is resolving. And then Rais also showed that this cyclic AMP cascade could activate RAC1. This is a small GTPase. And this activation of GTPase activated the phagocytosis by the bacteria. So when you don't have part 2, there is a increased inflammatory cascade. Also, the macrophages are not able to phagocytize bacteria. So these mice were dying of pneumonia. And then Jagdish was working in the lab at the same time, and he showed that there is also another cascade in which their TLR4 activation leads to activation of an enzyme, which is called sphingosine kinase 2 in macrophages. And this sphingosine kinase 2 generate S1P, which is a sphingosine 1-phosphate or bioactive lipid. And when you have this S1P, what it does is it blocks the STING activation. So STING is generally a C-gas dinucleotide pathway, which is emerged as a very popular pathway within last 10 years, activated by C-gas enzyme. And this STING then activate phospho-TBK in the Golgi, where there's phosphorylation of IRF3 that goes into the nucleus and makes this FN beta. So generation of this C-GAM, NF-kappa B, phospho and fat has to be controlled to really promote the host defense, but then resolve the injury. So when you don't have this S1P generation or cyclic AMP, the outcome of the resolution is not achieved, and thereby leads to death of this animal. So we are thinking how to promote these two cascades in the lung. So what I will be describing is some of them is published, but some are in the bioarchive, still in the process of publication. And this is totally unpublished and unsubmitted story, which I would like to receive your feedback on these signaling cascade. So what I will describe is that how signals that can regulate cyclic AMP instruct lung macrophages, and what is the role of RGS2 as an immune checkpoint in macrophages, and the role of E-type receptor, which we just discovered in last couple of months, and PKA axis in regulating afrocytotic function of macrophages. So let's see the role of the signaling cascade that keeps cyclic AMP in check. So what we did here, Ian Ratchford in lab was a graduate student in the lab at that time. So he used a LizMGFP mouse obtained from Classley, and he used very acute model of lung injury in which he had these mice inhaled LPS, and then he tried and he traced the lung macrophages, both cyclic F and CD11B positive. So you can see initially these macrophages are all cyclic F positive, as we'll imagine. They are pure alveolar macrophages, and they remain up to 4 hours. But when your injury starts to develop, there is also shift of these alveolar macrophages, and they acquire both the lineage, cyclic F as well as CD11B. And he showed that there is decrease in the pure population, which is cyclic F, AM, or alveolar macrophage. And at the time, he just kept it common, like interstitial macrophages or more derived macrophages, and these macrophages increases at that time. And then he showed, as you can see, that when these macrophages are transiting, there is injury, as you can see over here. And then within 24 hours, it resolves. So this model allows us to address what's the role of these incoming macrophages in the lung. So then what he asked the question, what is the mechanism that previous postdocs find cyclic AMP that keeps it in check? So he did the RNA-seq of the macrophages during injury and repair. And what he found was that phosphodiesterase 4B was the enzyme that was highly expressed in these macrophages. And it goes further on during injury, but then come back to the same expression level when the injury is resolving. And he proved through many experiments that this enzyme was very active in alveolar macrophages, not in the interstitial macrophages. And what he showed was then using roliprem, which we know inhibits PDE4B class of phosphodiesterases, to address the role of the cyclic AMP cascade in analysis of AM and lung injury. So what he found was that when he gives this roliprem after 1 hour of LPS inhalation in these mice, you can see the injury develops as you would expect. And these black dots are the Rolipram treated mouse. But you can appreciate that at 16 hours, there is a fast resolution period. So there is no difference in the Rolipram treated mouse at 4 hours. But at 16 hours, there is a very quick resolution of the lung injury, even in the mice. And what he showed was that it was cyclic AMP was induced at 4 hours, although we don't see the resolution at that time. But at 16 hours, we don't see cyclic AMP level were similar. So we think that there is some kind of a programming going on by the cyclic AMP release and Rolipram that keeps the injury in check. So to address this function further, whether these were alveolar macrophages or they were MOMAC, which were coming in the lung to resolve the injury, what he did was he used the CD11B diphtheria toxin mice, so which Jagdish earlier in the lab showed when you have deplete these CD11B macrophages using diphtheria toxin. So these mice have, so diphtheria toxin receptor is only expressed in the human macrophages. So this was genetically transgenic mice in which you can inject DT to selectively deplete these CD11B macrophages. So Jagdish showed at that time that when you deplete these macrophages, you get the lung injuries happen at the same level, but these DTR or CD11B null mice cannot resolve the injury. And with Pseudomonas, he showed the same thing, and these mice were dying rapidly after sensing the bacteria. So using these mice, he asked, okay, let me check whether it is monocyte-derived macrophages or it is alveolar macrophage population that is dampening the cyclic AMP level. So here what he did was he used just the monocyte which were isolated from LPS-treated mice. And first he isolated the monocyte, then he injected those monocytes in the mice which were treated with LPS or DT or LPS and no DT. And then he assessed the lung edema. So what he found was that when he had injected the monocyte in the DTR or monocyte depleted mice, the Rolipram was effective. Otherwise, if you don't have monocytes, you deplete it using this CD11B DTR mice, you don't see the effect of Rolipram. So what he concluded that these are the monocyte-derived macrophages that are important in resolving the lung injury in response to the Rolipram. And he further shows that if he use these Rolipram, he can reduce the INOS, which is a well-known inflammatory factor, IL-6 as well as IFN gamma. And he further showed that these Rolipram-trained AM were actually taking the E. coli beads, which were the phagocytosis. So both the cyclic AMP augmentation of the Gris cascade not only improved the edema, but also improved the alveolar macrophage phagocytic function. Thus, he then thought, okay, these are monocytes only, so it could be interstitial macrophages or monocyte-derived macrophages. So can we have training of these alveolar macrophages? So in another experiment, what he did, and along with Jagdish, what he did was, he treated the mice with the LPS, and then he treated with Rolipram, as in the first experiment, and then flow sorted these macrophages when one at 16 hours, because I showed you 16 hours, those macrophages were having trained and they were resolving the lung injury. And then he injected into a very different mice, which was wild type mice and hit with LPS. So what he found that when he has given no cell here, you as usual, this is your basal edema. But when you give them the LPS and no cell, the injury develops as we would have. So this is all wild type mice, but the cells are coming from mice that were treated with vehicle or Rolipram for 16 hours. But when he gave these roly-prime trained cells. You can see clearly that in the wild-type mice, these alveolar macrophages, we think, were actually resolving the edema. So that means adopted transfer of the cyclic AMP programmed alveolar macrophages, or more like macrophages, were capable of resolving edema. And he further showed that these were actually can be visible and SIGLAC positive and GFP positive, because these were the sorted from LISM GFP mice and SIGLAC positive. And in this paper, he showed actually what was inducing PDE4. It was NFAT again, and this phospho-NFAT had a binding site of PDE4B. So during injury, NFAT gets activated. So he was merging his studies with Rais, who showed NFAT augment NF-kappa B, and he showed this NFAT also augment PDE4B, with the net result of dampening cyclic AMP cascade. Thus, there was some kind of a yin and yang between calcium and cyclic AMP. Having seen this role of cyclic AMP cascade, we then moved into another aim, what is the role of RGS2. So if everybody knows, this RGS2 is a cyclic AMP response gene. It could be also responsive to other cascade, but mainly RGS2. So next I will describe this RGS2 role as immune checkpoint. So what is RGS2? So RGS2 is, we call it a GTPase activin protein. So if you have known the GPCR biology, so many agonists, such as I have listed over here, your angiotensin receptor, norepinephrine receptor, they couple to different G proteins. So these G proteins has to be inhibited after they have done their job of activating the signaling by the proteins which are called regulator of G protein signaling. And there are various flavors of RGS in the system, any system, your endothelial cell, macrophages. This RGS2 is highly expressed in the macrophages. And what is known about this RGS2 is that it is a GTPase activating protein for G-alpha Q. It has low affinity for GI, but mainly for the G-alpha Q. And it can inhibit the calcium, and that has been proven. So if you have RGS2 and GQ constitutive active, it won't work, because constitutive active GQ lacks the RGS2 binding site. So that's how it was proven. So based on this finding that cyclic AMP cascade was important, and RGS2 is a downstream effector of cyclic AMP, we looked over, okay, what is the expression level of RGS2 in different cell type? Oops. So first we looked over tabula muris, how much RGS2 was expressed in different cell type of the lung. And what we found that RGS2 expression was high, as I mentioned, in non-classical, classical monocytes, myelin cell, as well as alveolar macrophages. And some were also expressed in T cell, but very low in endothelial cell. And we then followed the published study from Mold et al., who showed in the JCI Insight paper that there are different population of macrophages that come during injury versus repair. So we thought, okay, what is the level of RGS2 in those macrophages? So day 3 is the injury macrophages. The day 6, they labeled as the repair macrophages. And you can see the RGS2 expression is decreased in the macrophages. Those were associated during injury, but during repair, in reparative macrophages, RGS2 expression increases again. And corollary, we have a NOS2, which is an inflammatory factor. So we compare the expression of RGS2 and NOS. So you can clearly see that RGS2 was going low during inflammation, but then coming back during resolution period. So having this, Jagdish again pursued on this RGS2 story, and he looked over this LPS now, rather than Ian model, where he'd used the acute model of reversible injury, he gives LPS intratracheal, which is a sustained model of lung injury, in which it takes about 72 or even 120 hours for resolution of lung injury. So he gave these just wild type mice to see whether what we bioinformatically checked whether RGS2 was expressed in macrophages. So he gave the LPS in wild type mice and then analyzed the lung injury and the RGS2 expression. So you can see that as we expect, lung injury goes, but in this case, it happens between 4 to 16 hours, and then lung injury resolved right here. And then he took out the alveolar macrophages using the bronchoalveolar lavage, whatsoever was coming in the lavage. That means you can have recruited macrophages, you can have resident macrophages, which are proliferating. And what he found, as we found during the bioinformatics, that RGS2 level in these macrophages by expression, the qPCR, decreased at 16 hours. But then when the injury was resolving, the RGS2 come back. And this is the protein data for the RGS2 that it goes down and then kind of coming back towards the normal level. Having seen that, then he got these wild type and the RGS2 null mouse and see how they react with this with intratracheal LPS. So he measured lung wet to dry ratio, which is the measure of your injury, as well as albumin influx into the lung. So what he found was that wild type mice, as we expected right here, they get injury, but they resolve. But in RGS2 knockout, so these are global knockout mice, the injury would not resolve. It is kind of sustained, both at the level of albumin permeability as well as the water accumulation. And he also showed that there were a lot of neutrophils in the lavage, even at this when wild type has barely any neutrophils seen, but in the RGS2 knockout, you have sustained influx of the neutrophils, and that remained over the time and leading the impaired resolution of injury as quantified over here. So having seen this, what he next did, okay, is it because these were global mouse, so one would have asked whether it is a hematopoietic RGS2 or is it parenchyma RGS2 in the lung that is resolving the injury. So he took those wild type and RGS2 knockout mice, did the sublethal irradiation and bone marrow transfer, wild type to RGS2 knockout and RGS2 knockout to wild type mice, and repeated that same protocol after 5 weeks. As you can see, the lung RGS2 was decreased right here, and was barely any expression in the parenchyma. When he gave the RGS2 back to the wild type mice, they behaved just like RGS2 knockout mice. But when he gave wild type hematopoietic cells to RGS2 knockout, the injury resolved, indicating that we don't have to worry about the parenchyma, although RGS2 is maybe lightly expressed in endothelial cell, but for the lung injury resolution, it is the hematopoietic cell, and we believe it's the macrophages that are playing role in resolving injury. So then we looked over these macrophages, which were coming in the lavage, to see what were the transcriptomic of these macrophages. And what we found, so we focused on 64-hour because we wanted to see what are the level of the gene as the injury was not resolving. And what we found was it was interferon gamma cascade was upregulated. So if you look at GVP4 was NOS2, interferon gamma receptor, IRF7, GVP, all GBPs were accelerated. So we wonder whether IFN gamma has a role in this process. So we focused on the estimating IFN gamma level in these mice. And we measured, so first we did in the lung injury patients with the help of Shaolin from China. So he works on ARDS patient. And when he sent us the data, with IFN gamma was very high in the lavage of the ARDS patient. These were very sick patients. Some patient resolves, some patient doesn't. And when he compared the RGS2 expression with IFN gamma, you can see there is no relationship between RGS2 and IFN gamma in the control patient. But when he does this in pneumonia patient, you can see the patient who is resolving, going back to the home, they have very low hyphen gamma level, but patients who were sick on ventilator, they have very high hyphen gamma level and very low RGS2 level. And similarly, we did in the SARS-CoV-2, COVID-19 exposed mice here. So we measure the RGS2 mRNA as well as hyphen gamma level. And as you can see that RGS2 level decrease in the mice where they had SARS-CoV-19 and the hyphen gamma was increased. So all these were associated changes, but kind of proven that the RGS2 has important role in resolving injury. Then we looked over how is the hyphen gamma level in these RGS2 knockout mice, alveolar macrophages. And we found that hyphen gamma level was very high in RGS2 knockout alveolar macrophages, while in the wild type, it was almost barely visible, but hey, oh, some problem here. So in 64 hours, so this is 0 hour, and the RGS2 null AM still have very high level of hyphen gamma. And he could show using the lavage of these BAL AM, and he could show that, and I think Charlene will be interested, somehow these are having some kind of a puncta, it's forming like a deposition of hyphen gamma as puncta. I don't know, trafficking is involved or what is involved, but Charlene may be interested in these findings, how this hyphen gamma are accumulated at vesicles and accumulated towards the membrane. And then he showed that the same thing if we do from BMDM of wild type and RGS2 knockout mice, we similarly find there is exaggerated level of FN gamma when we expose to LPS. These BMDM for 8 to 16 hours it increases, but see in the RGS2 knockout BMDM, it is hugely increased and doesn't go back as in wild type. And then what we found was that this, of course, was stat is known cascade, and we didn't find any lab in any changes in stat 2, stat 3, stat 4, stat 5, and stat 6, but it was only stat 1 that was showing high phosphorylation at later time points when this FN gamma was accumulating. And when we did the JAK inhibitor in these macrophages, we could see that this, so these are the wild type macrophages, and we do the JAK inhibitor, you can inhibit the phosphostat as you would expect. But in the RGS2 knockout, you see the persistent level of stat as you see on the control setting. But when you did the JAK inhibitor, you could inhibit the stat phosphorylation. So indicating these alveolar macrophages were secreting hyphen gamma, and that then ligate its receptor because JAK is downstream of that. So by inhibiting JAK, we could prove that hyphen gamma is working in a paracrine manner. And then we looked over, because RGS2 is a gap, GTP is activating protein for GQ. So we looked over what is the role of GQ under this process. So we focused again on hyphen gamma determination. Here, before that, we also generated this AM specific mice. So these are the CD169 So what we had the CRISPR-Cas9 because we couldn't find any RGS2 flox mice. They were in repository. So that means we have to breed them for years just to get the macrophage specific knife. So here we use this approach of CRISPR-Cas9. We bred with CD169 CRE, and then we injected guide RNA, which is RGS2 guide RNA or control guide RNA by LPS, analyze the lung injury. And you can see these RGS2 guide RNA decrease RGS2. This is lavage, but not the control. And when we estimated the hyphen gamma in these macrophages in the lung on white to dry ratio, you can clearly see the mice, CRISPR mice, CD169, which were getting the RGS2 guide RNA, they had very huge level of hyphen gamma in the lung at the basal state as well as during injury. So this is all done in 64 hours. And these mice were not able, so these are wild type mice, there is basically resolution of edema, but in this RGS2 knockout mice, you have basal edema as you would see over here, but then they would not also resolve edema when you have given the guide RNA. That means RGS2 in macrophages, probably we do not know they are totally alveolar macrophages, but these may be but this data suggests that alveolar macrophages has a role in RGS2 in alveolar macrophage has a role in resolving injury. Then we looked over if that is true, whether if we give back RGS2 in the macrophages by delivering the RGS2 cDNA, after we have given before giving LPS. So what we generally do, because there is no RGS promoter which we can drive RGS2 only in the macrophages. So this was a general cDNA given IV because these were more derived macrophages. So we gave the RGS2 cDNA or vector alone and hit with LPS after 32 hours, where we see most of the time the genes are expressed. And then we assess the lung injury at 56 hours or 72 hours. So you can see the vector, you don't see any expression of the RGS2, while when we gave back RGS2 cDNA, you can see RGS2 is expressed in the lungs. And when we estimated the wet to dry ratio in these lungs, we can see that mice which were receiving RGS2 cDNA back, they were able to resolve, but RGS2 null mice, which they received vector, they were not able to resolve. So indicating RGS2 has a role. So both CRISPR-Cas9 as well as this cDNA delivery prove that these are RGS2 positive alveolar macrophages or more derived macrophages that are resolving injury. And further, Jagdish show that with just to prove that these are expressed in the ball AM, he also did the RGS2 fluorescence. I'm not showing the two-photon image that RGS2 express in the macrophages after cDNA delivery. And it also reduced the FN gamma level, as can be seen over here. After that, what he did was, okay, what's the role of G-alpha Q? So we did first assess the inhibitor of GQ, which is BIM 46187, which binds the GQ pocket and inhibits the GQ activity. Very short. So we did the calcium imaging with this BIM inhibitor and proved that yes, it was using thrombin. We showed that it was inhibiting the calcium signaling in macrophages as well as endothelial cells, so proving this inhibitor works. And then we did this inhibitor experiment in the macrophages. So these are the control macrophages, and these are the wild type macrophages, and these are the red line, dashed line treated with the BIM, that means GQ inhibitor. So you can clearly see that even in the RGS2 knockout or BMDM, there is huge level of alpha gamma as I showed earlier. But with the BIM, it reduced to almost nothing as equivalent to what we see in the wild type macrophages. And this BIM also reversed the alpha gamma generation even at the later time point, saying as GQ cascade got upregulated in these macrophages due to the absence of the RGS2. And then we gave this inhibitor in vivo. Nobody has done so far. This GQ trial in the mice. So these were just wild type mice. And we gave LPS and the BIM inhibitor as well as the RGS2 null mice. And then we analyzed, and there were papers published using GQ inhibitor for some other disease where they have given 3 milligram per kg. So we gave this inhibitor and analyzed the lung injury. So at first we assessed the FN gamma in the lung, and you can see that wild type, even in wild type lungs, the BIM brought back the FN gamma towards the very low level. And surprisingly, in the RGS2 knockout mice, that BIM brought the FN gamma level equivalent to what we have, even lower than the wild type, indicating GQ somehow is generating this FN gamma mRNA downstream of RGS2. And this is the wet-dry ratio with the BIM to prove that these two are correlated inversely, that RGS2 is suppressing FN gamma. And if you don't have RGS2, GQ activity makes FN gamma. So if you inhibit GQ, you inhibit FN gamma, you also resolve the lung edema, as you can see over here. So we compare the wild type 64 versus 16, you can see there is inhibition of the edema formation. And the RGS2 mice, you can reverse that edema in the presence of the BIM, but not in the vehicle control. Then we compare the G-alpha-Q activity inhibition. What is it doing, the AM transcriptome? And surprisingly, this inhibitor reversed what we saw, the FN gamma cascade earlier, the transcriptome. The GBP4 was the highest expressed protein in the macrophages. It got low. NOS2 got low. Another GBP binding protein, FN gamma R, FN gamma R2. So basically this study, what he showed was that there's a normal gradation of your G-alpha-Q activity during LPS, and we do not know how G-alpha-Q is working. That is the open question right now. But what we think is that when you hit the macrophages with the LPS, there is a very low grade of FN gamma generation by the TLR itself. But when you don't have RGS2, this GQ pathway dominates, it secretes FN gamma, it makes FN gamma that then go back to, in a paracrine manner, activates the FN gamma receptor, which I think it's increased as I showed you, and lead to phosphorylation or phosphostat and inflammation. So when you don't have RGS2, you kind of have uncontrolled innate immune response and non-resolvable lung injury. So next I will discuss the recent pathway which Dr. Banerjee in the lab, a postdoc, is following on this cascade. So what he's trying to link is that how this cyclic AMP pathway has a role in regulating the afrocytotic function of the macrophages. So we know that when you injure, as I showed, there is a lot of neutrophils in the lavage, which can be seen at 16 hours, 72 hours, and they resolve in 120 hours. So we did the head-on comparison, how it goes between more-derived macrophages and BAL-AM. What's the level of these macrophages in the BAL when PMN are coming? So we can see that very little MOMACs are there when there is high neutrophil accumulation in the lavage. And these MOMAC increases in number as the lung is resolving, as neutrophils number is going down. But we also see that as I mentioned, AM number decreases, but then AM number increases during resolution. So the question was, which of these two macrophage population is helping to clear the neutrophils in the lung during injury? To address this function, what we did was, we did the afrocytotic assay. So what he did was he injected LPS in the mice, wild-type mice, and after 14 or 70 hours, so this is ex vivo afrocytosis, he gave the apoptotic cell. So he made this apoptotic cell by exposing them to the heat and quantifying that they are really dead cell. And then he does the BAL analysis after 2 hours. And what he found, so this is a complicated FACS plot. So here he segregate the cyclic F and CD11B, but are taking the apoptotic cell. So what he found, the AM take around 6% of apoptotic cell, while your MOMAC take 14% of apoptotic cell. And this is the quantification at 16 hours. And when you go beyond 72 hours, where is right here, you can see again the AM is taking 1/3 or what MOMAC is taking. So both at 16 hour where you have injury, maximum injury, mostly these PMNs are being cleared by your MOMAC. But even though the number is so low, there you can clearly see that this MO derived, which are coming in the lavage, they are clearing the neutrophils. So then he used these ketchup mice in which these ketchup mice, the neutrophils are red in color. So he exposed these ketchup mice to, so we call it an in vivo or endogenous apoptosis. Then ketchup mice, he hits with LPS as intratracheal and then perform the ball and see how many cells are red there and which are taken by these macrophages. So he does the FACS plot sorting of this, and he shows that the apoptotic cells are taken again mostly by the MOMAC as compared to the alveolar macrophages. This is at 16 hours and this is at 72 hours. So both in ex vivo apoptosis assay as well as endogenous apoptosis assay, he shows that these are the MOMAC which are being recruited into the lung during injury. They are performing this apoptotic function. When he looks over, okay, where this distinction is attributed to these two population of macrophages, that alveolar macrophages are there to begin with, MOMAC comes during injury, where this distinction is attributed. So then he goes to in vitro studies where he makes the MO-derived macrophages using the regular procedure like MCSF, or he makes the GBDM or alveolar macrophage-like cell using TGF-beta and GCF. And after 7 days, he compares and analyzes them. So what he sees, the apoptosis, is that he used these two types of cells, and so he called them as MBMDM or GBMDM. So these are all isolated from the LIS-MGFP mice and programmed using MCSF or GMCSF and TGF-beta. So he compares the apoptosis at 45 minutes and 90 minutes. And what he sees is again these MO-derived macrophages or MBMDM are taking these apoptotic cell faster as compared to GBMDM at both 45 and 90 minutes. He then performed the FACS analysis again and again shows that you can see a big bubble over here as compared to GBMDM. The apoptotic cells are being taken by the MBMDM as compared to GBMDM. And we thought, okay, maybe these GBMDM or MDMDM has different type of apoptotic receptor that are describing or that are giving these abilities for differential function. But if you see our typical apoptotic receptors and the ligands such as GAS6, S1PR1, TIMD4, they are highly expressed in MOMAC, and TM1, AXL, K Taro, they are also expressed in EM. So we don't think it's the apoptotic receptor that are giving this differential regulation of apoptotic ability by these two types of the macrophages. So to understand the mechanism, what's going on, he looks over the mitochondrial membrane potential because R.A. and others have shown that when you have apoptotic cell engulfment by the macrophages, there are changes in the mitochondrial membrane potential. So he uses these in vitro derived macrophages at 1 day to 7 day and estimate the TMRM, which is a macrophage membrane potential. And you can see the membrane potential of these macrophages barely changed. It's stuck to the same level, and I think it's moved here. But when he compares the same thing with the GBMDM or alveolar macrophage-like cell, you can see that this 1, 3, 5, 7, the potential increases hugely between 5 and 7 days as compared to MO-derived macrophages. And he doesn't see any changes in the mitochondria. So he did the mitochondrial mass. There is no change in the mitochondrial mass, but only the mitochondrial membrane potential. So he estimated the ridges, he estimated the length of these fragments. There is no change. So something else is going on in these mitochondria that has led to low membrane potential in the MBMDM than in the GBMDM. And he then do this comparison in vivo. So what is the TMRM looks like in vivo using classical monocyte, non-classical monocyte, MOMAC, and AM. And again, find that MOMAC has, and monocyte and MOMAC, they all have very low mitochondrial membrane potential as compared to alveolar macrophages. And similarly, he compares the plural alveolar macrophages with other macrophages, which are generally regulated using the MCSF environment. And again, he sees that plural or LPM macrophages have low membrane potential as compared to alveolar macrophages. And so I asked him, is it because of the apoptotic cell? Are they more hungry? Why your mitochondrial potential is changing? So he comes back that it seems like apoptotic cells are regulating the mitochondrial potential only on the GBMDM, but not in the MDMDM. Somehow these macrophages, which are generated using the MCSF, they are born with low membrane potential. But you can program the GBMDM to have low mitochondrial potential after exposing to apoptotic cell. So then the question is what is regulating this membrane potential in these type of macrophages that then is linked with probably afrocytotic function. So he looked over the GO pathway based on the RNA-seq he does, and he found again that this adenylate cyclase activating GPCR signaling pathway has emerged as the highly regulated pathway in these macrophages. And then he estimate the pKa, which is a protein kinase A, and as the end stage of the cyclic AMP dependent enzyme. So we have a colleague who has a sensor, and he's very focused on the cyclic AMP pathway. So we gave the macrophages to him. He made this measure he has this sensor which reports about pKa activity, and he comes back, oh, your MBMDM has high pK activity as compared to GBMDM. And then Som again with the collaborator Zahid measured the phosphorylation of this pKa and again find the GBMDM has barely an pKa activity as compared to MBMDM. Now we know that this pKa can be activated by COX enzyme, and when we compare the COX enzyme level in BMDM or MBMDM, you can see this enzyme COX2, PTGS2 is highly expressed in the MBMDM as compared to GBMDM. So then he got the COX floor sensor and measured the COX floor activity, which will report for the PTGS2 or COX2 activity. And again, he found the COX2 activity is much higher in MBMDM than the GBMDM. And we know that COX2 activation leads to synthesis of PGE2 that then binds this receptor, could be EP13 or EP24, that then leads to cyclic AMP and pKa. In the case of EP13, it blocks cyclic AMP while PGE2 binding of its EP24 receptor activates cyclic AMP synthesis. So we wonder, is this, if we provide PGE2 directly to this, and if you remember, there were a couple of papers from Aratab's lab that he hinted about this PGE2 role in afrocytosis. So we then provided PGE2 directly to the macrophages, and we found that PGE2 actually can augment the afrocytotic function of GBDM. So these are no PGE2, and if you give PGE2, the apoptotic cell uptake is increased. And similarly, if you inhibit PKA using H89, you can actually increase the afrocytotic function gets impaired. But then when you provide PGE2, you can override that function. That means PKA pathway somehow and mitochondrial potential are linked together for the afrocytotic function. And then he compared this mitochondrial potential, the membrane potential in presence of PGE2 after seeing that PGE2 is altering the afrocytotic function of GBMDM. And you can see clearly that PGE2 supplementation barely changed mitochondrial potential because it's already so low. But when he gives this PGE2 in GBMDM, you can decrease the mitochondrial potential to the level of MBMDM, like here, the blue. And this is the quantification he can do with the same thing with the H89. Given PGE2, you can reduce the mitochondrial potential, and that is reversed to the GBMDM if he gives PGE2 or H89. And he shows this on the imaging level also using the mitochondrial mass sensor as well as mitosox as well as TMRM, that PGE2 supplementation decreases. These are all GBMDM. You can reduce the mitochondrial potential, and then when you inhibit the PKA, the mitochondrial potential comes back. And these are the quantification of that plot. And he also looked over, is it possible that DRP, which is GTPase in the mitochondria, because is it that involved? And he just used the MDB1, which is a inhibitor of the this dynamine like GTPase in the mitochondria, and he doesn't see any effect of MDB1 on PGE2 augmentation of the mitochondria potential, indicating that this is not linked to the DRP function, rather it is some PGE2 regulation of PKA and then some alteration in the mitochondria that is governing the mitochondrial membrane potential. So then we asked the question, okay, can we add the EP4 agonism? So he tried various combination with the EP2 receptor agonist and EP4. He came back with the EP4 as a more promising receptor that promotes the PKA and cyclic AMP generation in the macrophages. So first he measured the COX-4 activity in vivo, in MOMAC, AM, and EPI. So this is all in vivo. And he finds that MOMAC again in vivo has a most COX-4 activity, and you can see more also in epithelial cell, but not much in endothelial cell. Then he gives LPS into the wild type animals, and then gives PGE2 or H89 and analyze the ball to see what is the level of afrocytosis by EP4 agonism. So first he tries with either H89, which inhibiting PKA, or activating PGE2, PKA pathway. And he shows that when he inhibit PKA, he reduces the afrocytotic capacity of the macrophages, alveolar macrophages. This is done at 72 hours. But when he puts back PGE2 in presence of H89, it doesn't work. It still have low, it's more increased. So that means PGE2 bypass the afrocytotic function of the alveolar macrophages when PK is inhibited. And then he tries this mitochondrial potential, same is still he is doing it. He can also reverse the mitochondrial potential in AM using the PGE2. It's right here, and then it can reverse. And he tried then the PGE2 and H89 and then looked over the now PMN clearance because that was the ultimate goal. And he sees that when he gives PGE2, the clearance of the PMN is faster, even at 72 hours, when that, as we showed earlier. But with H89, when you inhibit PKA, the PMN clearance decreases. And similarly, this Rubenprost, so this is a agonist for EP4, and this is GW, which is inhibitor of the EP4. You can see that the apoptotic cell uptake by the AM increases by Rubenprost as compared to the inhibition of this EP4. And BAL PMN also decreases grossly compared to your GW when you given the Rubenprost. So indicating that EP4 agonism has a role in promoting AM function in terms of afrocytotic capability. Now the question is, if that is true, can we train AM for more afrocytotic function even in the absence of your MOMAC? So here we turn our attention to CCR2 knockout because CCR2 has low number of monocyte per ml of blood. So we thought, okay, this is a good mouse to ask the question whether we can train AM using AP4 agonism. So here, this is a broad data where we just compare the PMN in the lavage of wild type and CCR2 knockout mice during LPS-induced lung injury and as we repair. So you can see that in the wild type mice, as we saw earlier, the neutrophils are very high at 16 and 72 hours, but then go back. But in the CCR2 knockout mouse, which is these bars, the neutrophil number remains high at 72 hours and very, very high at 120 hours. And then when he gives the Rubenprost in these mice at this time point, 72 hours, where he see the differences, he could reverse this number of PMN. So that means by activating the EP4 receptor in AM of the CCR2 knockout mice, he could train this AM function of afrocytosis. So the model emerges that MOMAC, when they get infiltrated into the lung during injury, they provide the PGE2 for AM to train the afrocytotic function. So they are more afrocytotic, the MOMAC, but then they also release the PGE2 to train AM for the afrocytotic function. But when you directly give this PGE2 activator or EP4 activator, you can promote the afrocytotic function of the alveolar macrophages. So I stop here and I will take the question and any suggestion on this project. Thank you.